Hello.
Hey Serena, how are you? There's no chemistry? Like, what the heck? Like, as a topic?
What do you mean there's no chemistry?
There's no chemistry.
Where? I thought
Let me try it.
the
Okay.
abstract
No,
was about dipeptides and stuff.
no, I mean as a topic to like add it here on Clubhouse.
Oh, I see.
What the heck?
I should complain about that.
Yes. Hi, Ankit. I don't know. I see you joined today and that you're member of the Science Society. I'm following you because you're most likely in Dr. Ulin's lab. So feel free to come to the stage. So how's it? Hi, Eric. How are you?
Hello, Eric.
I'm all right. Hello, Serena, Katerina, everyone else. Happy Thursday. Uh, yeah, nice weather for me. Looking forward to learning some new crazy science.
Yep.
This
Me
looks like
too.
it could qualify.
It's a whole new institute here in New York that he opened up. So it's really cool. Yeah, I'm really looking forward to this talk. So. But that is so weird that on Clubhouse there's no topic option. Chemistry.
That is weird.
Well, chemistry is just complicated physics. Let's be honest here.
Or is it more fundamental physics and biology?
But I
We're
can.
always in a weird place because, whatever we think of physicists, we know the biologists think of us in turn.
Yeah, for at least my university, the feud was more regarding engineers, mathematicians, and physicists. So each of the professors of those departments kind of trashed the other departments, and it just made for a very unpleasant kind of atmosphere. And then when you actually work with teams that are multidisciplinary or disciplinary, you end up seeing that actually none of that stuff matters. So it was quite an experience.
Hey, Ryan. Welcome to our club. Thank you for making it.
Hello.
Hello.
The unmute button is all the way on the bottom right corner. The little microphone symbol.
Got you. Can you hear me now?
Oh,
Yep.
yeah.
Perfect.
Loud and
Oh,
clear.
hi everybody. Sorry about that.
Oh,
Hello.
no worries.
Welcome.
Thanks.
I
Excited
hope.
to be here.
Yeah, we are excited to hear about your research. Is Ankit part of your lab?
Yes. So Ankit is, and he said, I think he's there. So
Yep.
Ankit is there, and I think Scott. Ankit and Scott are the two key people who did a lot of this work. So Scott, I don't see yet, but he said he would join us from Scotland, actually, where he is right now.
Oh, wonderful.
Oh,
So hey,
wonderful.
Ankit. So
Sorry, go ahead. I'm sorry.
So I see you made Ankit part of the. He's been moved to speaker.
Yes.
So that's great. So Ankit can help me explain the story. That's excellent.
Perfect. I don't think I have a bio from you, Ankit. So I apologize if I did.
I can introduce him if you
Yeah.
like. I know Ankit pretty well.
Okay.
So.
Okay, I'll introduce you. You introduce Ankit. So that's perfect.
Perfect.
We'll start in around 4 minutes.
Yeah.
That's okay to give people time to arrive. Yeah.
I'm just going to plug my phone in to make sure that we don't run out of battery midway.
Yeah, Clubhouse drains the battery, don't you think, everyone? Like, Clubhouse is a battery draining app.
Well, I'm never far from a charger.
That's a true Clubhouse addict. Yeah, me neither. That's good.
Well, I'm scanning the slides. This is going to be so fun.
Right, they are so pretty.
Well, it's actually a nice opportunity for us because we've had this story. Ankit and I are super proud of it. We've worked on it for about 5 years, and it's complex, but it's also kind of relevant, we think, to a lot of things. So we've been trying to figure out the best ways to present this to people who are not experts. So this is a really nice opportunity for us to see how that goes, if we can make sense out of all the complexity.
Well, great. My background's chemistry, and I have
That's good.
I have a deep pet theory about the origin of life. So I'm already seeing
Okay,
stuff.
that's
That's really
funny.
cool.
Yeah, we've been trying to avoid making big claims about origin of life, but on the other hand, we do think there is some relevance there. So we can definitely talk about that.
Okay, good. But I also did work in computer-aided molecular design, so I
Okay, it's right up your street then.
yeah, this looks great.
Yeah, in this work we didn't use much computer-aided any. In fact, this was all well informed by biology and then purely experimental. But we also do use computation a lot, actually, more and more so in our lab, to help figure out some design rules and things like that. So yeah.
Well, great. I can't wait.
Yeah, it's really exciting. I'm a former biophysicist turned entrepreneur who works on a sensor for rapid ID of pathogens. And we're always thinking of how the next generation of not only diagnostics, but also synthesis could perhaps be aided by new perspectives in either environment or processes. So looking forward to it.
Yeah. How are you doing, Ankit? I think you can unmute if you feel comfortable. So, right bottom corner, I just learned.
Okay, Katerina, think we could get started, or should we?
Yeah, let's Ankit find the unmute button. It's like a little microphone symbol on the bottom right hand corner. If you press on that, we can hear you. Maybe he's away from the phone right now. Okay. Then we can.
Okay, we'll figure it out. I can. Yeah, that's.
If he is having a trouble with the interface, it may help to close the app and restart it. The classic, have you tried turning it off and turning it on again? As is the case with most apps nowadays. Sometimes they get a little quirky. So.
Okay. Okay, then let's start. Welcome everyone to the Science Society. Oh yeah, Ankit wrote in the chat. Sorry, trying but can't join right now. He's restarting it. Okay, he will be back. Good. Welcome everyone to the Science Society. We are very honored to have our guest speaker here today, and I hope I'm saying his name right, Dr. Ulijn. And let me give you a little bit of background information. He is the founding director of the Nanoscience Initiative at the Advanced Science Research Center at CUNY in New York. And he did his master's in biotechnology at the University of Wageningen in the Netherlands, and his PhD in physical chemistry, University of Strathclyde in the UK. And he has held several personal fellowships and has won a number of awards, including the RSC's Norman Heatley Medal and Royal Society Merit Award in 2014. And his lab is focused on figuring out how the molecules and processes of life can be engineered and repurposed to develop new materials and technologies that respond to their context and are green and sustainable. These approaches have wide-ranging applications, ranging from energy harvesting, tissue engineering, sensing, and new ways of chemical information processing. I think this is quite amazing and important work. So thank you for being here. And if it's okay, one of us will ask you a more general question. Serena, do you want to go ahead and
Sure.
ask?
So we usually like to start with a very general question just to get to know you a little better. Was there any point in your life and growing up that you realized that you're going to become a scientist, or what was your first experience that really led you down this path?
Oh well, thanks. That's a great question. So yeah, I remember exactly actually when I decided that I probably wanted to be a scientist. When I was probably about 14 years old, in my biology class in high school, I learned about genetics and I learned that biology, with all its complexity, can be logical and predictable. We learned about Mendel genetics. Then later, I learned about the Watson-Crick double helix DNA, and that all of this is encoded into molecules. And that to me was incredible. I still think that's incredible. And I've been ever since been very very interested in figuring out more about how biology works. And then I guess when I went to university, I liked the idea. So then this was early 90s, when biotechnology just became a thing. So I was in the Netherlands, and there was this new course that you could study in biotechnology, which basically was the promise of using what biology can do so amazingly for a variety of applications that may or may not be explored already by biology. So going essentially beyond what biology can do with biology's tools. So that really intrigued me. And then I guess I learned that to really have a deep understanding, you have to understand the molecules that are responsible really well. So that's when I made the shift to chemistry, physical chemistry, and then ended up in materials and nano. But yeah, it's been, I guess, an ongoing. I've been driven by curiosity and hopefully doing something meaningful also, and being I'm very glad that the job exists, of being a scientist and a professor, because it's a great job. So I'm very glad I went down this path.
Well, it's fascinating. There's so many similarities to what drew me down to the molecular level as well. Um, I'm curious then, having you paint the picture of just reaching for more and more fundamentals. At what point did your curiosity bring you to the present work, where it just looks like a fascinating presentation ahead? But what drove you here?
Yeah, thank you. I guess the realization. So at some point, you learn more and more about how living things work, and you quickly come to realize that it's incredibly complex, and that we still have very 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 limited understanding of how biology does its engineering. And then I guess I like the idea of I guess a reductionist approach there, where you can basically break the whole thing down into simpler pieces that you can study and can understand and can predict. So we've done that. That's actually been my research focus really until maybe a few years ago, where then we started to understand the pieces pretty well. But using a reductionist approach, you also limit yourself really, because biology is inherently not reductionist. So now what we started to do is these simpler pieces that we now understand, we put them back together again in different ways to build complexity. So that's kind of how it worked, I guess. So realizing that biology is incredibly complex, then going reductionist to end up with something that you can engineer and understand, and then building complexity back in, but from those simple pieces. So it's actually different from what biology does. Biology does complexity from pieces that themselves are also complex. So you look at one protein as a very complex, object or machine, and then within a cell you have hundreds of thousands of these things working together. The building block, the individual pieces are complex, and the collective even more so. So we really asked the question: can we simplify the building blocks but still have biology's complexity approach? So yeah, that's how we got here. And I would say this paper, that's why I'm really flattered that you guys picked it up. For us, it's a real first step into complexity after that reductionist approach. Yeah, I hope that
Okay,
makes sense. Yeah.
wonderful. All right. Well, so the audience can access the paper at the link in the top, and we
Yeah.
can follow along. And so take us away.
Okay, great. Thank you. I'm going to just reposition myself here so I can see I hope I have enough battery life now. Because I think if people can see the presentation, that is probably quite helpful to look at that. I mean, hopefully I can make sense also just by listening. But so I'll guide you through the slides, and I'll try to mention when I'm switching slides so people can follow. So the first slide is I just couldn't resist to put in a picture of our amazing building. So I was, as you heard, in the UK for most of my academic career, and I moved to New York 8 years ago to help set up this interdisciplinary research center that you can see here. If you're familiar with New York, we're actually in Harlem. We're looking down towards Midtown. You can see a sliver of green maybe in the middle. That's Central Park, and on the very right hand side you can see the Empire State Building, so as landmarks. So we're north of Central Park, so tourists don't tend to go there so often, but I guess I recommend if you like science and you like you like visiting New York, and go a little bit further north. There's a lot going on there right now. We have a wonderful building, wonderful community doing cool research. And yeah, the title of my talk is Building Complexity in Biological Design Spaces. So that's what I guess tried to explain in my answer to the previous question. Complexity is inherently hard to understand. Our view is if you build it and you understand all the pieces, then you can hopefully gain some understanding of how to engineer complexity. So instead of observing complexity and trying to figure out how it works, we're building it piece by piece from molecules, and then see how new behaviors emerge. So if you go to the next slide, so complex systems are mysterious in many ways. They're all around us, and they are a really huge science challenge, interdisciplinary science challenge, to understand how complexity and connections give rise to new behavior. So I show there 3 examples at completely different scales. Cells clearly composed of many elements working together, interacting, and collectively giving rise to behaviors. The middle image is the brain, and the right image, of course, the planet. And all of these have in common that they are complex systems, and they have an ability to encode and store and process and employ functional information. I took that line from the Santa Fe Institute, which is a very famous institute for complexity science in New Mexico. So understanding any of these objects in across the time scales and across the length scales, and understanding how they respond to new situations is incredibly interesting but incredibly difficult. And what we're trying to do is basically apply the same idea to chemistry and building it, as I said, from the bottom up. So complex systems, if you strip them down to their key components, they have elements, and these elements have interactions, and collectively this gives rise to a function or purpose. So if you think of the previous slide, you think about cells. Well, the elements could be organelles or proteins. The interactions could be, how these proteins interact with each other, and then the collective function or purpose could be something like sensing or movement or anything that a cell can do. It can do these things because all these elements work together. And then if you look at the middle picture there, so if you're in PowerPoint mode, just go click so the next thing comes up. These things then become stimuli responsive, in other words, responsive to their context, by essentially modulating their interactions. So if you put a system under any kind of stress, it will respond by changing the interactions between particular elements. And then if you go one step further, you get adaptation, where in this case the color changes from white to red, and that's to signify that if you as the system interacts and responds to its environment, in some special systems the elements can change. So the elements change their properties, and that gives rise to overall adaptation. And that's what basically happens in cells and in the brain and in planet Earth, that you have elements interacting collectively, they respond to their environment, and then the elements change as a result, and the overall things changes. And you can apply this to many other things. I mentioned the Santa Fe Institute. They also apply this to the economy, and basically anything that's composed of multiple systems and multiple elements and interactions, and responds collectively. Okay, so that's a lot of complexity there, but I try to break it down into something that's understandable. 6 elements that interact and collectively give rise to behavior. Okay, now I'm going to tell you how in this paper we believe we have achieved basically a chemical system that does the 3 things that are shown on this slide. So if you go to the next slide, I'm going to make the connection with biological design spaces. So our complex system is built in a biological design space, but with elements that are minimalistic. If you look at what we understand currently of how proteins work, proteins are composed, of course, of 20 amino acid building blocks. So on the left-hand side, we see biology's toolbox. All proteins are composed of these 20 building blocks. For those of you who are chemists, you will recognize that these can undergo different types of interactions, the ones in purple versus the ones in green versus the ones in blue, in red. So they basically have interactions programmed into them, and quite simple interactions. But when you put these together in long chains, like in proteins, this becomes very, very complex. And if you think about the cell, the elements in the cell could be proteins that are themselves quite complex. But that complexity is now fairly well understood in that we have artificial intelligence, like Google's AlphaFold has made a lot of news over the last couple of years in that they are able now to predict, and this number is dated, it's probably over 1 million now, protein structures directly from the primary sequence. So in other words, you put 100 or 150 or 200 or 300 or 1,000 of these amino acid building blocks in a string. This folds up spontaneously, and this is now predictable. It's also possible to engineer proteins and make them better, make them do different things using tools like directed evolution, which won a Nobel Prize for Francis Arnold a few years ago. And it's also increasingly possible to engineer this through computational design. So that's all wonderful, and it means that proteins to an extent are understood. But on the other hand, it's also really not very well understood, because what we cannot do, everything here is based on what biology has already come up with. So all the proteins that biology has come up with, we can basically now understand and predict how they fold. But from the amino acid building blocks, there's other design spaces that have not been explored by evolution, and it is very likely that you can build really interesting machines and materials by in that kind of design space. So we don't have any tools yet to explore other design spaces. And other things we can't yet do is do complexity, as I just explained it before. Proteins are understood, but how proteins then interact and collectively give rise to new behaviors, that's a frontier that's still very difficult. So our research is focused on essentially forget about proteins, but going straight from these amino acid building blocks to make small elements, elements not in the chemical sense, but in the system sense. So and then essentially design complex systems. So if you go to the next slide, so I'm now on slide 5. This is what this looks like. So what we have now is the elements that are going to interact are dipeptides. So they're combinations of 2 amino acids. The first one is responsible for interaction. The second one is responsible for aggregation. So that means that we have dipeptides, small molecules that have 2 features built in. One is to aggregate and the other one is to interact. And what we've done in the experiments I'm going to talk to you about are to mix 5 of these together. So you have 5 different molecules that have different interaction propensities. You mix them together, and then that becomes our system. And then later on, I will show you that we've gone a step further and actually mixed 15 of them together to make a complex system. And the complexity goes beyond that, but I'll show you that in a minute. So what happens? So imagine these 15 molecules that all have different ways programmed into them to interact. If you mix them together, you get self-organization. So if you look at in the diagram on slide 5 and on the right-hand side, you can see now that all these dipeptides, the different colors signify different types of interactions. So blue is positively charged and red is negatively charged. So you can see some of those interacting. And you can see that the light gray, dark gray, and black, that's the aggregation propensity. And in the center, you can see that some of these are now interacting. So in other words, you have all these molecules, you put them in a solution, and they themselves start to organize themselves. So that is essentially the first, you basically have everything in place here to make a complex system. You have elements and that will interact and give rise to collective properties. Then the next step is that we challenge this system. So we can, for example, change the temperature, or in this case, we actually added ATPs. So that's, biology's energy currency. We add that to that system. So you have this mixture of molecules all interacting, and now we add this other ligand molecule. And this ATP molecule has itself, properties. It has charge, it has negative charge, phosphate groups. It has hydrogen bonding interactions shown in green. And then it has this purple piece, which is aromatic. So if we mix that in with our 5 or 15 elements, we can start to see that they now organize themselves around that ATP to basically minimize the impact of introducing this new molecule. So I hope that makes sense. So you're introducing a new molecule which has a lot of opportunities to interact. So as a result, the peptide elements organize themselves around that. So maybe I should stop here briefly because I'm going to go to the next step, but I'm afraid I may have lost 1 or 2 people already. So if there's anything I can clarify at this point, or shall I just continue?
All good for me. Anyone have any questions?
I was just curious. When you say organized, is it like kind of like based on like charge versus like polar versus non-polar? You know,
Yeah.
charge versus. Okay, okay.
Exactly. It's that. So so there's hydrogen bonds, there's hydrophobicity, there's charge, there's pi interactions, aromatic interactions. So all of these together will give rise. So it's basically the same drivers that drive a protein to fold, but now we don't have one long chain. We have lots of small pieces, and they organize themselves around this ligand. Yeah, that's a good question. So okay, so we have an experiment, a test tube in Eppendorf, where now these things are organized. I'm just showing you cartoons right now, but I'm going to show you some data in a moment. So the next step is we now we now try to achieve this. We now try to achieve that the elements can actually themselves adapt and change in response to the challenge. So we do that by now adding an enzyme called endoamidase. And what that is basically is an enzyme that can make and break bonds between peptides. So remember, our elements are all these little dipeptides that can interact in different ways. So I'm on slide 7. So if you look in the top corner of slide 7, there is again the cartoon structure and then the chemical structure of one of the dipeptides. And then you have this endoamidase, which is an enzyme that can connect these dipeptides to form longer peptides. And that's a reversible reaction. So we can basically make and break peptide bonds so that we go from dipeptide to tetrapeptide and longer. So I'm only showing tetrapeptide, but it can go longer. The key now is that it does so, it doesn't do so normally. It's not favorable energetically to spontaneously make a peptide bond. So in principle, the equilibrium of this reaction is to the left-hand side, where you basically have all the dipeptides. However, if the molecule on the right-hand side, the tetrapeptide, somehow binds to something or aggregates or is in any way stabilized, the equilibrium can shift to the right. So what I'm saying here is that we now introduce a reversible chemistry into this system, where if the dipeptide doesn't interact and is not part of the system, it doesn't do anything. But if it becomes part of the system and is binding to something, it can basically make more complex, larger peptides. So we add this catalyst, and we can now make new sequences. So I hope that makes sense. So that's all the pieces that we use. So what that looks like then is that, again, here's my cartoon. So this is still slide 7. You can see that some dipeptides are not really involved in the system on the outside. They're not interacting with anything. There's one in the top left corner. There's one in the top right corner. There's one kind of in the middle. And then in the center of the image, you can see that some of the gray-black aggregating pieces start to assemble. And as a result, we can now start to see that connections are made using that enzyme between the peptide bonds. And you can see here formation of a hexapeptide. You can see that 3 dipeptides are binding together, and it's driven by interactions with each other and with the ATP. So I hope, so this is kind of the idea and the hypothesis. Can we make something like that work? So then Ankit comes in, who's spent 5 years basically making this happen, and with help from this gentleman, Scott McPhee, who is an exceptional analytical chemist and who has helped us to figure out what's going on. I should say for the chemists among you, you'll be very familiar with this. Chemists have a problem with mixtures. So chemists prefer to work on pure compounds, and also chemists tend to have a problem with heterogeneity. Chemists tend to prefer to work in solutions. So if something doesn't dissolve, we have a problem. If there's too many molecules in your sample, it's a mess. So this is for chemists quite a challenge and a change to try to look at how many molecules in one system work together instead of studying one molecule. So the way we do this is so Scott basically looked at the mixtures that we end up with, and he looked at the formation of these tetrapeptides. So as I said before, everything on the left is stuff that doesn't interact and doesn't do very much. And then on the right, the tetrapeptides form only when there's a reason for them to form. So you can see on slide 8 things that I won't go to in any detail now, but basically you do chromatography. So you do your messy experiment, you do chromatography, and then you use computer-aided analysis to basically pick out all the individual tetrapeptide peaks that are formed, and then you can basically start to map what's going on. So what you see in the bottom right corner on slide 8 is intensity maps, intensity fingerprint essentially of all the possible peptides that can form, and you can see that some of them are dark blue, which means they form because they interact. Others are white, they don't form much at all, and others are somewhere in between. So this gives you a readout essentially of the complex system that we've been working with. So Ankit then did a number of very interesting experiments. So he started with a set of 5 dipeptides. The aggregation propensity of them is low, so they have the white piece of the colored dipeptides means low aggregation propensity, and we then have these different colors. We introduced the ATP and the endoamidase, and then basically how to read this is in blue are the first dipeptides, in black the second, so overall making up the tetrapeptide. And you can see in the absence of ATP, there is a certain distribution that forms. And in the presence of 100 millimolar ATP, you can see that there is now upregulation of the upper right corner peptide, which is GR. And you can do a differential map where yellow basically means downregulated and blue means upregulated. So we have a readout now of tetrapeptides that form and ones that don't form. And then he did the experiment again, and then he got a different result, as you can see here. So on slide 8 still, you can see a different distribution of blue. The blues are slightly less intense, but there are more of them that light up. And that is, in the beginning, we found that very puzzling, but later on, we actually learned that this is inherent in a system of this level of complexity. There are so many possible interactions that the system can find multiple ways to solve one problem. So in other words, we add ATP, and this system is able to compensate for that in multiple different ways. So it has multiple solutions to one problem. So that's not something that you would want or see very often in chemistry, and it's something very typical in biology, this idea of a stochastic response. So this is our first piece of evidence that we are kind of navigating a space that is somewhere between chemistry and biology in terms of complexity. So then he moved on, so this is slide 10, to a system that also has assembly or higher aggregation propensity. So you can see now the second amino acid is black. In this case, we use valine instead of glycine, which has a level of assembly propensity. So that means that we have interactions with the ATP, but also a tendency to form structure, to self-assemble, to form structure. Now, if you look at this from left to right, days 1, 2, 3, 4, 6, and 8, with ATP, you can see over time clearly that there's a pattern kind of growing in. While minus ATP, it kind of stays the same over time. And then you look at the difference map, the blue, yellow, and you can see even more pronounced that there is a cluster of peptides that really are amplified in this case. And we can follow that over time. So this is now on the right-hand side of slide 10. In the absence of ATP, you can see peptides form. So there's 3 shown here, but in total there's 25. And you can see that they kind of stay, the levels stay the same without the ATP. But with the ATP, you can see the green one goes down, the pink one keeps going up. And that means that it is basically recruiting more and more material to make more of itself. And it's starting to essentially, it's not exactly a replicator, but it starts to make more copies of itself, that VK-VH peptide. And ultimately, that leads to formation of actual objects. So the kind of fuzzy microscopy image that you see before is taken from a very complex mixture. That's why it's not the most beautiful microscopy. You can see a lot of these little dots, these little objects appear, and they in turn are nucleation sites to make even longer peptides of specific sequence. So what we're doing here is we start with these 5 dipeptides that themselves have some code inside them, but as we assemble and over time we build in more and more complexity. So the self-organization leads to sequence selective enhanced oligomerization. So we don't tell this thing what to do, it figures itself out and starts to amplify formation of certain peptides. And this is spontaneous. So, there was a comment made earlier about
Quick
origin of
question.
life relevant. Sorry, did someone have a question?
Oh, did I even interrupt the origin of life? No.
Yeah. No,
Quick
go ahead.
question. The length scale on the microscopy. What?
Oh yeah, sorry. That is. I think that is most likely a 100 nanometer scale bar. I think
Okay.
these little objects that you form are in the nanoscale. Yeah.
Nice. Okay.
And then the last thing I'll show is the most complex experiment we've done, the slide 11. Now we use all 15 dipeptides. So we have a mixture of 15 dipeptides with 3 different aggregation propensities. Mix them all together, add the ATP and the enzyme. The first experiment looks at a difference in temperature, where you can see, so on slide 11, you're now looking at 225 tetrapeptides. They're all formed, we can detect them all. They're all interacting in this little reaction vessel. And as you can see, by comparing the experiment at 2 different temperatures, we can clearly see a gradient from yellow to blue, and that gradient goes with the aggregation propensity. Then the one next to that, the map next to that, actually goes shows you what happens in the presence and absence of ATP. And you can now I can see it clearly because I've looked at this so many times, but hopefully you can see that there's little blue islands form. And that there's also an overall gradient. So again, the overall gradient is from yellow in the top left corner to more blue in the bottom right corner, but also you see very clearly islands form. So these patterns here are essentially, I mean, they're not QR codes clearly, but they're a readout. And it's not digital. It's not like 1, 0. Each kind of pixel has information over the amount of peptide that's formed. And this is a unique, so every situation will give you a unique like fingerprint or readout, and that's what we think is pretty exciting about this. So then if you look at the this is actually going to appear in the paper is in press right now. It will be in the June issue of Cell Press chemistry flagship journal Chem. And so we heard that we will feature on the cover, which is kind of exciting. So the picture you see on the right hand side is kind of an artist impression of our experiment. And for those of you who paid attention in the beginnings, we're based in New York, so this is meant to we put like a little central park inside there to make this look like the city we love, but it's also a representation of the up and down regulation of these individual peptides. So with that, I do hope that I haven't confused everybody. The next steps, we think that this is the start of something very interesting. We've shown the structures, we've shown the beginning of structures that can respond to changing conditions through sequence adaptation. We've shown the beginnings of using this as complex sensing. These complex mixtures will give out a different readout depending on whatever you throw at it. So it is a unique fingerprinting approach, even though it's complicated. It ultimately, the readout should be easily interpretable. We're thinking about things like chemical memory, because we are building we're building in a similar way, in some ways, to how memories are created by interaction paths. And if we repeat the same paths over and over again, we think we can do something with chemical memory. So the idea is, you take biology's building blocks, the amino acids, essentially forget about what biology did with them, but restart the whole journey, and do chemical and functional information processing to make new materials. So the last slide, slide 13, is acknowledgement. So that's our team. You can see Ankit standing actually next to me. You can also see there's a cake on the table, and that this was actually Ankit finished his work in my lab a few months ago. So this was his leaving party. And then there's the funders. So that's what I have to share with you. So very happy. I don't know if Ankit is able to unmute to help me with Q&A. I see he's back. Happy to discuss. And I see Ankit is unmuted, so I think I'm going to have some help with the Q&A. So thank you, everybody.
Yeah, thank you so much for this really amazing presentation. Such a amazing basic science work. Like that is so interesting. So Ankit, welcome. Do you have something to add that you would like to share first? Okay, please flash your mics and go ahead with questions, everyone. Thank you.
So my question is, it seems like a very stochastic process that's very exciting, because I think that means there's a lot of potential for synthesis. You briefly mentioned that there was multiple solutions to any one problem. What was the kind of rate of divergence, or however it is that you quantified that kind of process? Because I'm thinking, we throw a bunch of things into a test tube, and then we set the right initial conditions and boundary conditions, and perhaps there's a certain sink or source of either heat or energy, as in the case that you did with ATP. So I'm just very fascinated with kind of the richness of that complex space, but it seems like a very treacherous space, and it seems very unforgiving. So if you could comment on that briefly, if possible.
Yeah, no problem. I mean, I think the best, we have done something we actually wanted to start to put a number on, and essentially repeat the experiment, not like 3 or 4 times, but maybe 20 times, to really get a feel for how stochastic is this, and are there 3 solutions that can be binned, or are there really an infinite number of solutions? So what we've observed, we've done, of course, publishing in a nice journal, we had to make sure that every experiment was done multiple times. So if you look in the supplementary information of this paper, the last experiments that I mentioned, where we start with 15 dipeptides and end up with 225 tetrapeptides, the overall patterns look very similar. So that the idea of the gradient and those little islands coming up, But if you look at the detail, it is really actually remarkably different. So, you know, you can look at this in two ways. So you can focus on the fact that so if you follow an individual tetrapeptide, you can see things that are quite different. But the collective, the overall pattern that comes out is very, very similar. So we actually talk in the paper about robustness, and actually we say that these systems are robust in that they can essentially solve a problem in multiple ways that give a very, very similar ultimate outcome, if you see what I mean. But yeah, if you're looking for a system that will give you precisely the same result every time, this is inherently not it. And it's exactly, you know, when you try to teach a chemist to do biology, this is exactly the frustration that you know, cells don't ever do exactly the same thing, and that's because of the stochastic nature. So yeah, I think it's as you nicely summarized, it's a feature of the system, and it's good and bad at the same time. I guess it makes it robust in a way, but it does mean that you are unlikely to find the exact same result twice. I think biology is like that. And again, if you want to make the connection with the origin of life, you know, having some variability in how you can solve a problem is probably a key thing to, you know, adaptation and biology. Ankit, do you want to add something to this?
I think Ankit is posting in the chat because his microphone is not
Oh,
working.
okay. So I can't actually see the chat, but
Yeah,
oh, I can see the chat. Yes.
you can.
Yeah. Right.
So,
Sometimes,
thank you very much.
Ankit, sometimes exiting the app and then re-entering it may help
He
with.
tried already. Ankit, are you maybe at the university? Because sometimes when you use the Wi-Fi from a public space like a university or hospital or so, they kind of block some features from social media. So if you use your cell phone data plan, it usually then works. We had that before with some guest speakers.
Yeah, and just to make sure that we're not missing obvious things, the bottom right hand is the unmute feature, and if you're wearing potentially a headset, that might not be great either.
Does this work?
Yes,
Sorry.
loud and clear.
Oh,
Finally,
Yes,
the famous
very good.
sorry,
Hey,
Ryan.
no problem,
And sorry,
Ankit.
guys.
I'm glad you're here to, you know, it's going to get difficult from here on, I'm sure, as the questions get hard. So you're just on time. Nice to hear you.
So, yes, to answer the question, Ryan actually summed it up nicely. And as I mentioned in the comments, the stochastic nature is unforgiving. And that is one of the things that was actually not observed in, I should say, the traditional chemical origins of life, in the sense that if you start from simpler components, you are never going to get very definite answers. This is just the reality of the landscape, which is something what we observed and had not been observed before. And what that forces us to see is that when the components are small, that means if the stochastic nature is higher, you look at the global components of the ensemble and not sort of home down on singular components. And that's why what we do is that of the heat maps that Ryan showed, we do a principal component analysis on it, so that each heat map is essentially a single point on that analysis, and it tells us how one mixture is uniquely different from the other. This is how we separate mixtures essentially. So that sort of works. And yes, it is unforgiving, but that is essentially the reality and something what chemical origins of life had to go through as well, most probably.
So to follow up on that, is it likely that, because I think one of the hypotheses that I always found controversial was, you know, the tree of life, that there was one common ancestor. I always tend to be in the basket of, yeah, there's probably many bushes of life. And I'm not sure if your work supports that, but if you could comment on how the nature of the origin of life perhaps pertains to this.
Um,
Yeah. So Anke, do you want to give your take?
Yes. So
Go for
the last so that common ancestor is essentially last universal common ancestor. That's called uh, the the kind of chemical origin of life that looks at is actually much before that. So last universal common ancestor is then that particular bacteria or cell is actually life at that moment. This looks at, or our work or work similar to such as this, look at times much before that, in which there was stochasticity, but that sort of got amplified and concentrated into very few sequences, very few functional sequences. So yes, the Luca or the uh, last universal common ancestor could have been actually not that diverse. Uh, Again, that is obviously up for debate because that sort of research has not been done. But for certain, the previous mixtures, something that was the makeup of life, they were certainly much more diverse. And they went through this concentration mechanism. And something that Ryan explained as well, in which what we actually see is that dipeptides form tetrapeptides, form these nanostructures, which then magnify similar hexapeptides, essentially. So what's happening is that once a functionality is honed in, it sort of is amplifying on its own, like the some sort of stuff that looks like itself. So did I answer the question?
Yeah. This kind of reminds me of work done by Lee Cronin, where his molecular assembly kind of addresses one point of pre-selection, that it wasn't just all natural selection, it was a lot of pre-selection bias in the geochemistry or the other kinds of chemistry that existed beforehand. So this is interesting because it seems to suggest that all the building blocks kind of converge, and that's very fascinating because I think that's very hopeful for the rest of the universe. So maybe we have neighbors nearby.
I wanted to add that we had Dr. Sergei Krasnokutsky here from the Max Planck Institute for Astronomy, and he presented his recent paper of how peptides in space are formed on stardust. He experimentally showed this through condensation of atomic carbon. It's really, so he said basically it wouldn't be really special if peptides would form on stardust. Serena, you were here in the room, so yeah, it's
Yeah.
really interesting.
Yeah. Oh, go ahead, Dr. Ray.
Yeah.
Oh,
So,
go ahead.
so I think since you mentioned Lee Cronin's work, I mean we actually were inspired by a paper that he published in maybe 2 or 3 years ago now. That actually shows that you can do reversible condensation reactions and make peptide mixtures that become different dependent on their context. And I think in our case, what we can show, I think we have the first example here of actual formation of specific sequences. So out of all the possible, we showed that 225 tetrapeptide sequences. Some of them are amplified, other ones are not. But it gets more interesting because the ones that are amplified then in turn amplify formation of hexapeptides of specific sequence. So we start to see spontaneous formation of sequence. And that is clearly key to origin of life questions where making finding amino acids on meteorites and finding short peptides is one thing, but then actually going from there to longer sequences that then have eventually function and folds, that is another challenge that's not clear exactly how that will have happened. And I think showing that this can happen spontaneously purely based on things like interaction and aggregation and reversibility is interesting to us. But yeah, it definitely supports that origin of life is clearly messy and heterogeneous. But out of that mess, at some point, some order needs to emerge, and I guess that's what we're we're looking at. I mean, I'd like to say again, our objective is not to figure out the origins of life, but we do feel that there is some parallels there, definitely.
That's awesome. Yeah, Lee's work is definitely inspiring to many people, myself included. So he's always up to some crazy new scientific model or something. In his work on molecular assembly, talked about this in principle. And I recall when I was an undergrad, we talked about the dogma of the stochastic nature of the emergence of life, but it was never demonstrated. So it's nice to see some of these, I guess, principles or things that we took for granted for a long time actually be either verified or not. And yeah, I'm just curious what other potential possible accommodations, although this would be empirically intensive. I think one of the things that Lee is doing is saying, okay, well, we have some data from another planet. How do those initial conditions and boundary conditions and other elements affect the possible things that could emerge on that planet? And could they go as far as our system has gone? So very fascinating work. Thank you for the last question. I promise. Thanks.
Well, what's so fascinating about this opening in complexity that you've presented is there's so many other knobs to tweak. There's used ATP, for example. There's all sorts of other structures that could be templates to aggregation. The nature of the enzyme, you could modify the enzyme and have it select for different ranges of chemistry. But what gets really cool is when you start to form these or run these aggregation experiments and condensation experiments in the presence of previous products. So that taking the spontaneous products that emerged in or resulted in one case, and have those present in slight variations of another. So in essence, you would find spontaneous quaternary structure, perhaps form, and wouldn't it be cool if you got enzymatic activity? But lots of really just cool variations to explore.
Yeah, I mean, to be honest, probably. I mean, it may be obvious to some. Our our objective initially was actually to make the whole thing fueled by ATP. So that's why we picked ATP as the molecule, the ligand of interest here. The idea was that, and I still think that's possible. We've just not achieved it yet in this first paper. But if the ATP then also gets, so you build up these peptide structures, they build up around the ATP, and then if eventually they start to hydrolyze the ATP, the whole system should revert back to the original equilibrium position. So you then have essentially an ATP fueled system that gains function and complexity in the presence of ATP, and as ATP runs out, it goes back to its ground state. So that was the objective. Now, the ATP hydrolysis, I think it's entirely possible to get enzyme activity to emerge in these things, but we probably need to do a few other things like maybe add some metals or rethink our starting library. But that would be a really amazing thing to achieve. So I think we want to, aggregation and assembly is one thing, but there are certainly other biological functions that you can think of that we can mimic. So I agree. It would be very exciting if we can use this approach and then a catalyst emerges that amplifies things further. Ultimately, we'd love to do this whole thing without the actually adding an enzyme and get the system to figure it out itself. I mean, it in principle should be possible, but we're not there yet.
I had a question. Dealing with like I guess the whole enzyme. I think it was like endoaminase or something like that, right?
Yeah.
So my question is like you said that what you guys saw was like when you add the enzyme to the solution, it had the peptides and the ATP that like you saw the enzyme reaction shift to the right. So I was wondering like do you know if that's like due to like maybe like the lowering of the activation energy needed for that enzyme to go to the right, or maybe if it's like the organization caused by the ATP, like sterically put the peptides in a a way where the enzyme is more effective to.
Yeah, that's a great question. So I guess what you're asking is this a kinetic effect or a thermodynamic effect, or so why are you able to shift the equilibrium from left to right? So yeah, we call it endoamidase. I mean, it's a protease, but we selected a protease that does not like to cleave at the end of a peptide chain. So that makes sure that our dipeptides stay intact, because otherwise the thing becomes too complex to analyze reasonably by mass spec. So by using an endoamidase, we can basically connect and disconnect dipeptides. So but it's yeah, but normally amidases hydrolyze peptide bonds. So you would not necessarily expect, when you mix dipeptides together and you add a protease or an amidase, you wouldn't necessarily expect to find oligomerization. You would expect, hydrolysis. That's what proteases do. But that equilibrium position is actually not so far from, equilibrium constant of 1. So making and breaking peptide bonds is energetically, the equilibrium position is not so far removed, between the dipeptides in this case and the tetrapeptides. What makes it favorable to hydrolyze, and the reason why proteases hydrolyze peptide bonds, is mostly the ionization of the products. So you've got the amide bond, which is not charged, and you hydrolyze it into an acid and an amine, and at neutral pH that becomes highly ionized, and that basically drives it to hydrolysis. So but that is a reversible reaction. But when the tetrapeptide in this case is stabilized by self-assembly, or by binding to ATP, that basically adds a free energy contribution. And if you think in terms of like a free energy landscape, it would lower that well in the free energy landscape, which means that now becomes favorable. So in other words, peptides are hydrolyzed by proteases. However, if the peptide is stabilized, it can actually go the other way. That's what we're showing. So it's not really to do with the enzyme itself being, lowering, the enzyme lowers the activation barrier for sure, as enzymes do, but there's nothing special about the ATP or anything like that. It's basically the free energy of the precursors versus the product that you play with. I hope that helps. It was a very good question. I'm not sure if my answer was very clear.
I'm a little confused, but I'll think about it.
Yeah, I mean, I think what might help. So we published a paper that I think will help the understanding, where you basically start with dipeptides, and you show that if the product, the oligomer peptide product, has a tendency to self-assemble, you can basically drive this reaction all the way to the peptide. However, if the peptide doesn't have a tendency to self-assemble, it hydrolyzes. So essentially, self-assembly dictates whether you get condensation versus hydrolysis.
Gotcha. Thank you.
I had a question. First, thank you for your amazing presentation. Going through it, I was confused by the concept of stochastic rather, that being random, and versus the idea that under certain conditions things are not random. I'm confused. What am I missing?
Anke, do you want to take that one?
Yes. So, uh, ra- uh, let me just put two things in this context. So, stochastic nature that we observe is, for example, we mix 5 dipeptides in a reaction, and there is ATP in the reaction. And the products that come out are always, so ATP is actually negatively charged, and the products that actually come out that get amplified in almost all of the cases are positively charged, or in some cases they are with aromatic, but they are still positively charged. So stochasticity in our case is the relative concentrations of the positive charge systems. So we always see that the products that are amplified, they're always positively charged, which means that these are products that bind to a negatively charged ATP. However, what we see variation in, and that's where the randomness sort of comes in, is the actual concentration of these tetrapeptides. So what we see is, like, for example, sometimes, and sorry for mentioning some chemical amino acid names here if you're not familiar, but for example, there is aromatic amino acid histidine, and there are these positively charged amino acids, which is lysine and arginine. So what we see sometimes is that you have higher percentages of combinations of aromatic and positively charged amino acids, but sometimes there is a higher concentration of just two positively charged species. However, in the overall ensemble, the mixture is still that binds to ATP. So the randomness and the sort of convergence is these two aspects, in which there is convergence in the function, in which ultimately what's actually converging is the positively charged sequence, but what might vary is the kind of sequence that is getting amplified. That's where the randomness or stochastic nature is. Did that answer the question? Sorry.
Yes, yes, absolutely. Very clear now. Thank you.
Yeah, just to add to that, because it's really fascinating here, in the sense that the progression of the reaction affects the outcome, in the sense that if certain peptides are going to aggregate, they alter the free energy landscape of what can happen. And you can, I mean, it's a very complex space, but the there's there, in essence, the history of the evolving, the progression of the system changes that landscape, because it makes available different aggregates and different assemblies that may have impact in what's likely to react next.
That is a really very, very accurate comment. Thank you. I think that's exactly what, and therefore, you can. So history and time is going to be really interesting in this, because you probably noticed that the data that I shared, we had tetrapeptides, increasing in their concentration over the period of 8 days, and for these experiments, it wasn't really reasonable to go beyond like a week, but you can clearly, there's questions about what happens if you keep doing this. And I think the comment was made previously also, you can start to seed other things, right? So you have control here, so you can add other things over time and basically see how this thing continuously evolves and develops more complexity, and gets rid of essentially things that don't interact. So it is I think it's a system that can also lead us to just unexpected things that in over longer periods of time, it may well be possible to make things that are significantly more complex. I think the most complex. So Ankit, remind me, the longest peptides that you found of specific sequence, they were more than 10 amino acids, I think, right?
Yes.
So I think eventually you can really get their new functions and even folds, because these things are too short to fold. In that case, they don't have any secondary structures, but eventually they'll become long enough and they fold, and they start to do new things potentially.
It'd be interesting, and of course, you could sort of think of ways to perturb and try different systems here. But one interesting variation might be in terms of oscillating the pH over a long time scale to maybe as aggregations form, what's left on the surface may be more exposed, or it may change things, and in possibly altering the wet dry cycles in essence, to drive either favor condensation or favor hydrolysis. But there's just so many things to do with the system. It's fascinating.
Yeah, I mean, it's true. We have, and actually we still haven't exactly implemented that, but we thought that this would be, I mean, the complete hydration dehydration is interesting, but it also makes things, that's not really a reversible system. So I think in terms of the the ability to make and break amide bonds and not get any like unusual side chain interactions or anything like that. I think our preferred way forward is to continue to work in an aqueous environment, but I think things like thermal cycling can also be very interesting. So where you essentially use a PCR-like setup, you can just have a heat block that cycles periodically between high and low, and then basically you assemble, disassemble, assemble, disassemble continuously while you also allow for this condensation. I think interesting things will happen in a situation like that.
Have you start to look? I don't want to hog the stage. If there's any questions, please flash your mic or speak up. But in terms of, have you started to look to possible ways of functional readouts, or integrating, more type of sensors to into the system to interact with it and play with it at that nanometer scale, either optically or electronically, or any of that.
Yeah, I mean we have thought quite a bit about this. I mean there are, of course, the system doesn't have to be restricted to amino acids. We can actually, we have the liberty to do whatever we like, right? So it's not, because especially because we're not necessarily trying to solve origin of life questions. We're also trying to just create new ways of making adaptive materials. Simply the concept of having a material that behaves differently depending on its context can be pretty attractive. If you have to put a material system in a unknown environment and the material can adapt to that environment, it's a little bit like thinking about a material with a metabolism built in. That's another way of looking at this, besides the origin of life idea. So if you have a material with a metabolism built in, that material can express different properties depending on the context. And we've done previous work on looking at electronic properties of supramolecular materials, catalytic properties of supramolecular materials, optical properties like fluorescence, and those would all be reasonable readouts of this. So you could, for example, build in deliberately like a semiconductor component, organic semiconductor components in your peptide mixture, and then basically see how conduction emerges and how you can maybe make a conducting network that responds to its context. Or you can indeed think about a fluorescence readout where binding gives rise to reorganization that gives a fluorescence change. And as we mentioned earlier, like catalysis is an interesting one. So I think that's true, that also the impact of this work in the material science area will be that will come, I think, when we can show adaptive function. So there's many ways to do this. There's lots of ways to make functional structures based on self-assembling systems, and we can basically steal some of those ideas and build it into the system here. So that's hopefully what the future will bring. But we don't have any. We have plans, but no specific examples in that area yet.
I wanted to ask, I think, I'm not sure if I asked you before, if you know Dr. Marco Pettini's work where he showed this dipole long range electrodynamic intermolecular forces of up to 1,000 angstroms. I think it would be interesting. Like, do you think
Yes.
that these are the
Yes.
forces that
Hmm.
basically regulate these self-organizing properties?
So I'm not familiar with that work. Ankit, are you familiar with this work?
No, I'm not actually.
I can comment on it a little bit. It's a photonic effect that combines RF radiation and photonics to allow long range molecular interactions that would otherwise not form at the scale of about 1,000 angstroms. And this is interesting because the Earth has a magnetic field that's somewhat oscillatory. There's light energy that has some oscillatory functions. So for life, those seem to be two important recipes. And empirically, recently, they've shown that there is some compounding effect that promotes the forming of certain bonds if the effect is adequately modulated. So it was actually work that really fascinated us because I think typically people think of, oh yeah, RF, yeah, I'm not going to get coronavirus from 5G, but there's actually more to the story. It's more nuanced. Of course, we're always lifting the minimum floor energy, like the kind of minimum binding energies kind of increase with this extra additional energy, and it helps form some of these other bonds, but only when you have these two forces or sources of energy in tandem. And it was so fascinating. I had to reach out to, I believe his name was Marco, right, Katerina? So very, very fascinating work. You should
Okay.
send the reference, Katerina.
Yeah, I send it in the chat and direct message to both of you.
Oh, thank you. Yeah, we'll take a look at that. I mean, it sounds interesting. I mean, I have to have a proper look at it before I can say anything meaningful, I guess. But it sounds. I mean, any externally applied field or so we really believe that these systems that we've developed here they'll basically respond to anything you throw at it, whether it's chemical or physical or waves, and I think all of that is interesting. So I'd like to take a look at that work, and
What's really cool to add to this work is what he found was it was the vibrational modes within these. Say you have your complex assemblies. They have vibrational modes, and to the extent that those dipoles will oscillate, what he found is for aggregates that will have these vibrational modes and have these oscillating dipole-dipole interactions, if the frequencies are much different, it's a 1 over r to the sixth, and it's fairly non-specific. But as the frequencies start to match, it becomes more of a 1 over r to the third. And that's where you get the long range attraction. So what the observable would be is there'd be a tendency for clusters of similar resonance frequencies to aggregate, which would be really cool in your system
yeah.
because it would drive further aggregation without an external force. These would just be the natural modes of the system that was produced.
Yeah, we're going to take a look at this. That sounds really, really very interesting.
Yeah, we found it very compelling. We're probably going to set up a verification experiment here on our end just to see if we can reproduce the same kind of data because there is like a high frequency laser involved and a few other things. So it takes a while to get all those toys together. But it's exciting nevertheless because it represents not only a new phenomenon, but also, you let your imagination go wild and you think of all these therapies and interventions that could result or ways that we could stabilize disease. So it's very exciting work.
Yeah, please, if anyone has more questions, please go ahead. Dr. Shah, you didn't have a chance to speak yet.
Yeah, and thank you so much, Raina and Anki. That was fascinating work. And my question, um, I mean, back to the human body, I mean, not that much further about the dipole. So, because when we want to think about that, there is a different type of modulation that we can use it. For example, one of the modulation is the electrophile modulation that we can think about that about the selectivity and drug design. But what is interesting about your work, I was thinking about kind of adaptive or training the immune system based upon your work, because I see this capacity in this work, and I was just wondering, did you think about that or not?
So let me make sure I understand your question. So you said training the immune system.
Yes. I mean, well, for example, you know that we have two types of the immunity, innate and adaptive. So in the adaptive immune system, as long as you came with the assembly, self-assembly, based upon whatever you mentioned. So there's supposed to be optimization, right? When
Yeah.
it's happened, it means that there is a optimization happen. But you didn't mention, for example, in a human body or in vivo, we are talking about what part? Are we talking about, for example, tight junction, gap, or cell membrane, or where we can use it? That was just my question out of curiosity.
Yeah, I mean, I think we're quite, even though we of course use the same building blocks that the immune system uses. So there is I think a connection there down the line, and we are able to scramble, sequences, and our sequences should select for binding, which could be an antigen. So I think there could be a connection there. I mean, in fact, one of the reasons why we started this work, Ankit will remember this very well. In the beginning, we were actually hoping to find single unique sequences. So instead, we ended up finding, systems that collectively adapt to like a ligand like ATP. But our original plan was to use this setup to identify single peptide sequences that bind, that have an affinity for anything you throw at it. And I think that's still a possibility for this. But the systems level idea is I think then it becomes a little bit different from how I guess the immune system would tackle this. So there's a similarity in the fact that we use chemical information to find ligands, but in our case, it comes from many molecules working together rather than unique sequences or emerging over time. So I think there's a connection, but as a way to perhaps as an alternative or a way to find antigen binders. I'm not sure if this is going to outperform antibodies anytime soon. But it's an interesting thing to think about, like how the 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 collective chemical complexity does become a unique uh, binding situation, like an antibody. It's just composed of many different dynamic pieces in this case. So yeah,
As
I
long
like
as
I
we
like your thing.
can do, as
Yeah,
long as we can do deletion, we can do injection.
right.
There is both ways possible. That's why I asked that.
Yeah. Okay. Yeah, I think it's an interesting way to think about it. I'd have to give that some more thought.
I think this is very promising for any sort of synthesis that we want to engage in the future, because everything else is either labor intensive, either humans, or process intensive, and this is kind of uh, an interesting solution that kind of seemed like just a noisy place to be. So
Yeah,
I think it's very wonderful. I'd like to ask you more questions, of course, if possible. I'd like to connect with you and kind of shoot out some of my stupid questions, if I may, if that's possible.
of course.
Awesome.
Of course, I think the green chemistry connection that you're hinting at, I guess, is definitely a big driver for us. So this paper is one area of our research, but the general idea that you can make oligomers, that you can basically do polymerizations in water using enzymes and get specific sequence is in itself very, very promising. Because I guess we all probably agree that a bio-based future would be great, where most things are made of things that are inherently biodegradable and sustainable, and made from ultimately materials that you can produce from CO2 and light. So I think the green chemistry connection is definitely there for us. If we can learn how to really drive building blocks to specific products as a synthetic approach, I think it's going to be very promising.
It reminds me of recent Star Trek. They had something called programmable matter, and they had a very interesting kind of spin on that. So if you haven't already, check out Star Trek Discovery, or I have to also promote the recent one, Strange New Worlds. Both really good. I often find scientists are in the lab and working on something, and sometimes it's hard to see where this could be applied. So one question that I think that is kind of an ending question in terms of the applications. The kind of question that I think there's this person who worked for the government. They went from lab to lab, and their job, his name escapes me momentarily, so my apologies to the individual. But he would go to the lab and he would ask grad students and professors, where do you see this work in 70 years? So if I may, if that's something that you have thought of, if not, then perhaps another day. But if you were to see the long range limit, what's the kind of fantastical scenario that you see?
So did you say 70 as in 70?
Yeah, it's really hard to see that far. So even
Okay.
my criticism was also that. I was like, hold on a second.
Yeah, no, it's fine. I mean, I think it's nice to speculate, and I mean, okay, the dream. Well, the dream. I mean, so one thing that I often start when I talk to people about our, how our work can be relevant. So I really truly believe that you can make anything from biological building blocks. Anything. So you think about your cell phone, which is clearly a highly complex device. Wouldn't it be wonderful if you could make a cell phone just from fully biodegradable and sustainable components, i.e., a biodegradable cell phone where all the pieces are made of something that resembles proteins. I mean, with the proteins we know today, that's never going to happen because those proteins have been optimized for aqueous conditions. They don't perform they don't perform the functions that we're looking for. But if you take the 20 amino acid building blocks and basically say forget biology, and you actually start to evolve things in new ways that don't involve, for example, an aqueous context, it's unlikely that your future biodegradable phone will be made from components that are aqueous compatible. Actually, they probably shouldn't be. And we currently don't know how to optimize and evolve proteins that are especially suited for non-aqueous conditions. So that's one example where I think the types of the methodology we're developing, what we discussed today is all done in aqueous media. But you can do the same experiment in a non-aqueous solvent. So you take out the hydrophobic effects, and you actually start to make structures that are really potentially useful for use in non-aqueous conditions. So my point is that I think 70 years down the line, hopefully we will have worked out how to use biological space, sequence space, but explore completely different areas. Biology only covers a very, very small part of all the sequence space available in proteins. All the other parts remain to be explored, and they can lead to new technology, and they can ultimately lead to things like displays and electronics and everything else. So yeah, 70 years down the line, if I think it's probably essential actually that most things are made from something that's fully circular economy, biodegradable, and made from biocompatible building blocks. And I really hope that our methods can help, the design of these new structures. So yeah, that would be my answer. I don't know, Ankit, if you want to add something to that.
Um, so Ryan obviously gave a long-term picture of it, essentially answering the question. If I may, I'd like to give perhaps not a 70-year picture, but a shorter range thing uh, to it. So one of the questions, I mean, from the questions asked, and something that we've brainstormed as well, is that how to actually look at how to amplify specific sequences. Let's say, for example, one of the questions was that about antigen binding, or other functional questions as well, that would not require essentially an ensemble to work, but a specific particular peptide to be amplified. um, and we that was, as mentioned, was the initial goal of our project. But to brainstorm how to get there, uh, essentially what we have to do is uh, the polymerization process that goes from dimer to, let's say, longer peptides should not is currently stuck thermodynamically to tetrapeptides, hexapeptides, and you get diminishing yields of polypeptides as you go along. And that is one of the major hurdles that perhaps we would need to cross. If And that is something that Ryan's lab is working towards, is to cross it so that you get higher and higher oligomers to it. And one way actually to get there is actually to engineer the enzyme itself, so that that sort of is the restriction of the thermodynamic restriction essentially is driven towards a faster oligomerization process and a much more energetically possible oligomerization process. That is something I think would be a major breakthrough in our, and Ryan's lab is working towards it. Not exactly in enzyme engineering, but oligomerization of it. But that would be I think the next level of breakthrough in this work, which would allow our system to go from this fuzzy uh, stochastic regime to specific uh, sequence regime, in which you would, as mentioned, you could look at quaternary structures amplifying and such. So that would be for the material science side of it. I think that would be a challenge that I would look forward to, essentially, coming from Ryan's lab.
How very generous of you to pass on the work to the next generation. It's like this is left as an exercise for the next student. But yes, I agree. I think it's an interesting concept. I think the controlling aspect is definitely very difficult. But the person that we just mentioned a few moments ago, I think that represents an interesting avenue, because it's not introducing another chemical, but it's introducing perhaps a frequency-based control. And so that's really yeah. That's all for me now.
Yeah,
Another. Oh, go ahead, Ryan.
no, go ahead. Go ahead.
I was just, to pick up on a comment Dr. Shah made earlier with the antibodies. It would be interesting to, take an assembly that you find, and in essence, you can, you can cross-link it, say, so it's a stable coupled pile, but raise antibodies against it, in the sense that, and then conduct the reaction again in the presence of those antibodies. Would they in fact catalyze the amplification of what they recognized? That might be an interesting, because there is a precedent with catalytic antibodies where, um, but then this would be setting up a template of growing specific antibodies that would in essence provide a template that is already known to select for the assemblies that were previously observed. That's just a
Yeah,
thought.
that is it is an interesting idea. I mean, the catalytic antibodies, I think, it's a great concept that I think, well, as far as I know, never really completely delivered on its promise, in terms of, it's very, very hard to make anything resembling an enzyme. So transition state stabilization is clearly part of the whole puzzle, but the other part is really to get the induced fit to work properly. So when I think what you really need to make a good catalyst is a good balance of organizing because you mentioned they're cross-linking, and as soon as you cross-link, you kind of rigidify the whole thing as well. So there's a real balance between flexibility and structure that I think enzymes have just worked out perfectly, but very, very hard to replicate in a synthetic manner. And I actually think that I'm hoping that our system, because we give it so many building blocks, right? So what we focused on today was in the paper. If you remember, all the dipeptides have essentially two pieces: the structuring part, and then the interaction part. We've looked at glycine, alanine, and valine, and for those of you familiar with the amino acid alphabet, we can go essentially more strongly self-assembling, like phenylalanine, leucine. We kind of avoided that in this case because we wanted something highly dynamic. But if you go to those more strongly self-assembling pieces, I think it may be possible to ultimately end up with the right balance of flexibility and structure to get good catalysis. I mean, for us, it would be a dream to see a catalyst emerge from these experiments, and I think there's no reason why that can't happen. But yeah, we'd love to see that. And that may be, again, the catalytic antibodies was one. There's been a lot of interest in trying to achieve enzyme-like activity, catalytic antibodies being one approach. But yeah, we're curious to see if our approach can achieve anything there. I have to say, so far we haven't been able to do anything very convincing. But it would be a nice thing to show.
Yeah, quantification. I mean, when we are talking about the quantification, we have to, for example, think about insulin molecule. We are
Right.
talking about the couple of the peptides that they are just linked with the, for example, cysteine with each other. So cross-linking is undeniable in those kind of molecules. But whatever I'm thinking about is what organelle in a cell we should put it, because when we are talking about the structure, after that is going to be the function. And the function in a real cell happen under certain circumstances. It's not just, putting the peptide there and then amplify and then going through the process. That's why I just brought it up. So it was very fascinating work. Thank you for sharing again.
Sure.
Well, so if there's no other questions, I got a few more, or at least two.
Yeah, please go ahead. I'd love to hear your questions, Serena.
So in terms of tweaking the environment that these reactions that your system carries out, the class of compounds that we call dendrimers, if they come in all kinds of varieties, it would be curious to see what kinds of dendrimers might shape the energy landscape of your system, and in that case, have you thought about dendrimers?
No, we haven't, but I think it's interesting to think about dendrimers as like multivalent, presenting. If you think, I'm thinking dendrimers here with maybe their tips like functionalized with some kind of ligand where you basically get a lot more. You get a presentation of ligands in a multivalent way that I think our system would respond to in a very interesting way. Um, yeah, I think going beyond, we showed ATP, and ATP is clearly a small molecule. Going to more complex like templates, which could be a dendrimer, I think would be very interesting, and maybe also be the way to get to favor formation of longer peptides. Ankit, what do you think?
Uh, I agree. So dynamics is actually a very interesting idea. From the multivalent aspect uh, to begin with, but also um, because dendrimers, depending on their generation. I mean, if let's say a standard, let's say a generation 4 uh, dendrimer, if we go by, in which there are a lot of multivalent stuff present, but there is also a quaternary structure essentially, in the sense that the dendrimer molecule itself has a architecture, plus it has pockets. So point being that it would be interesting to see, like there will be, apart from the multivalent nature, there would be amplification based on these aspects of quaternary structure binding, or perhaps looking at hydrophobic pockets and that sort of amplifying it. That would be very interesting to look at, actually. We
Yeah,
certainly didn't think about it. I mean, one major reason being that dendrimers are generally very, uh, sort of slightly cumbersome to synthesize in an already complex system. However, it
yeah,
does provide a very unique challenge. I agree. It will be very fascinating to run libraries
yeah.
like
Even if the final generation were more of your dipeptides, so it would all be a peptide surface functionalized. But depending on which ones you used, you would have, sort of, you'd have these differential effects. So, that's that's cool. Another one, um, in this one goes back to, thinking through origin of life and what types of templates and aggregation structures would be present. I stumbled onto clays and the hydration clays, because they provide all kinds of environments. Some would select for the purines and pyrimidines and the gallies, and others, and the peptides would be more on the surface. But what I stumbled onto is the cosmetic industry has done an enormous amount of work with clays for delivery of cosmetics and pigments. And they have some very well defined chemistry in terms of these accessible tactoids, but these would be, smaller groups of the phyllosilicate stacks that you can control in solution. So it still would be an aqueous solution, and you can decorate them to give them more properties in terms of water solubility and so forth. But that would provide, certainly if you're doing peptides and nucleosides, the that would be some unique environments. And in some cases, they'll get into pillared clays where there's plenty of room for things to move around, but there's still stable structures. But that would be interesting environments that would certainly alter the aggregation effects. Just any thoughts of that?
Yeah, I mean, so clays have enormous surface area, right? So having introducing a interface, an additional interface like that, I think will be very very interesting. I mean, we I think we haven't really explored that whole idea of actually introducing a solid with a large surface area into the system, and well, it's very likely that you'll get um, preferential buildup of specific peptides at that solid liquid interface. I think clays are a great model system also because again they are, as you say, they would be, they're interesting for applications, and they're another green chemistry example. So we haven't done that, but it is pretty high on my priority list to start to have a look at what happens when you actually introduce and essentially a nano solid into these material into these systems, so you can look at what accumulates at the interface. So I think it's a very nice idea. But we haven't done anything with clays. I mean, the cosmetics connection though is quite clear to us that even though what we do is very much basic science, the peptides that come out of this have interesting structuring properties the cosmetics industry and the food industry is very interested in, simple short peptides that can, replace existing emulsifiers, for example, surfactants, gelators. So that is another area where there is actually applications around the corner. Because if you find, tetrapeptides or hexapeptides that can gelate water, or that can be powerful emulsifiers, they're low cost. They have low barriers to application if you compare them to synthetic polymers or any new chemistry. So I think that is actually if you wanted like quick applications, like not 70 years, but much sooner. That is, I think, the area where we should be looking. But yeah, I love the idea about looking for clay interactions. I think
Yeah,
that'll be
what was really cool is you could get, apparently in some cases, perhaps reagent grade, tactoids of known composition that are, and you can select for size, so like 100 nanometer, 200 nanometer,
right.
tactoids of, which would be just really, really cool to
Yeah, no,
characterize.
I agree. That'd
Yeah.
be very nice to look at that. Some very nice ideas coming up here.
Well, so it's been an hour and 40 minutes. How much more? We want to be respectful to your time. How much more longer do we have you for?
I can go for a little bit longer. I don't know about you, Ankit.
Me too.
Maybe. I mean, 9 o'clock or something would be. So that's New York time. So another 15 minutes or so, I can probably do.
Yeah, fair. So
Thank
that,
you.
yes.
Thank you so much. Yeah, please go ahead with questions. I don't know if Serena, you had probably more questions. Because
Well, I want to give other people a chance to have questions too. Or Ankit, I saw you on mic. Do you have more comments?
I just wanted to comment on the clay thing a little bit. So, as mentioned, yes, clay has obviously a huge amount of origin of life significance. And a paper I just wanted to highlight. There was a recent work, I think, a couple of years back, that came from Lee Cronin's group. I think it's a, if I'm correct, it's a PNAS 2019 work, which he actually looks at a limited range of amino acids polymerizing on a variety of clays. And he does not look at the molecular compositions of it, but he does principal component analysis to find out, to mention that different amount of clays, let it be montmorillonite or laponite or whatever, they give very different ensembles. Each of them actually give very different products, and that was one of the parts in that particular paper. I just wanted to highlight that yes, amino acids and clay really do go hand in hand, and there is something uh, good about, for example, a cosmetic function or something like that, is that Ryan's lab has shown that you really don't have to go to a very long length of amino peptides to actually get function out, at least for cosmetic purposes. You don't have to go that far to get function out of it. He's shown his other my colleagues in that lab had shown that tripeptides with 3 amino acids actually were enough to give you enough complexity for pretty cool cosmetic functions. So true. Just wanted to add to that earlier comment. Yeah.
I mean, the other thing is that clays are one of nature's nanostructures, and clearly there's many human-made nanostructures that, like graphene, carbon nanotubes, quantum dots, everything else, and finding short peptides that interact favorably with these so that they can be used for, say, biomedical applications is another area where I think what we're doing can be pretty useful. So we're trying to not restrict ourselves to things that are necessarily all biologically relevant, but also really look at this interface between non-biological nanotechnology and biological nanotechnology. So if you wanted to integrate anything, synthetic into a biological context, you need to think about the interface, and what happens at that interface is going to dictate probably how effective your, say, man-machine interface is going to be. So we think that our methods can be useful at identifying some molecules that could stabilize these interfaces and make that more seamless between the synthetic and the bio.
Yeah, I did some work on the hydration of clays that appeared in JAX some time ago. But during that time, it was quite fascinating to study the the edges of clays, because it was a particular simulation technique that allowed me to follow reactions. And but the edges of clays can support phosphate esters in the sense. And what's fascinating about that is the geometry of the clay layers nicely stacks three um, uh, nucleotide bases. So what that got me thinking about, genetic codes and things. It would be interesting to, if you ever do get into the clays, looking at triplets um, the um, of the nucleic acids and any particular preferential aggregation in the clays for the corresponding amino acids. Just leave
Okay.
you with that.
Yeah, no, that's we should look into that. I mean, we haven't really looked into this deeply, but I think yeah, it's something to definitely consider. So you said this has been done with nucleotides before?
Where did that person go? Hello,
Oh,
hello.
am I talking on mute? Okay, I was talking on mute. Sorry. No, the work I did with clays was it was computational,
Okay.
but it allowed me to take a number of on the side, it didn't make it into the paper, but on the side I would look at the geometries of clays and the geometries of these abiotic precursors to the functionality that we see in life today, and particularly associations of amino acids and nucleic acids, and where would the phosphates go? There were a number of fascinating coincidences of geometry. I can say it
Okay.
that
Okay. Interesting.
way.
Yeah, I think the geometry matching is. So we played around with I don't know if you're familiar with like ice binding proteins, and that basically, again, that's geometry matching. And I think all of those questions about how do you get a sequence that matches something, they lend themselves really well to this approach, because you basically just throw in all the building blocks and let the system solve itself. So we haven't really looked at a geometry matching question, but I think it's actually very, very interesting, and I think you're now kind of convincing me that we should look at that. And there's so many examples where you could look at like crystal structure matching or anything like that. I think that this system is well suited to something like that. So yeah, that's a great idea
Yeah,
actually.
I was gonna add
I
to
love
what
when we're
Serena.
able to.
Sorry, can I go?
Sorry, Ethan. Just a quick comment. I love when we're able to generate new homework. So that's always fun. Go ahead, Ethan.
Yeah, I was just gonna add to what Serena was saying, and maybe suggest as well, and even to is the do the same, but with the pyrimidines and purines rather than the whole nucleotide, especially since there's evidence in some of the primordial super experiments that purines and pyrimidines are actually created in certain amounts alongside the amino acids, like in the Milner-Urey experiments. So
Well,
that it might be easier to find. Yeah.
what's cool about the purines and pyrimidines and clays, because they'll get pulled right into the galleries,
Hmm.
into the inner layers, and that's part of what was so fascinating is they can the purines and pyrimidines will accumulate in the inner layers of the clay, and the amino acids will be more on the edges. But the phosphates won't. They don't have particular affinity for the inner layers. And during wet-dry cycles, they'll condense. Perhaps I'd love to see the chemical characterization of that. But they can estrify on the edges where they've got exposures there that they can bond to, particularly in dehydration conditions. And so amino acids on the edges, purines, pyrimidines in the inner layers. Isn't that wet-dry cycles? Isn't that a cool system?
Yeah, that's definitely cool. And then you need to find someone to analyze what's going on, and that's where it always gets very interesting. That's to us. I mean, this is really the key to doing this work. Would not have been possible without someone really committed to fix the analytical puzzle. Because the more things you mix, the cartoons always look beautiful, and it's nice to think up theories about how multiple collections of molecules interact and give rise to collective functions. But the analysis is very hard. So what you propose there is that purines and clays and amino acids, forming some kind of collective assembly. You can draw what that looks like, but how do you actually prove that it is indeed that organization? It's not. That's far from trivial.
Well, they've also, the pharmaceutical industry's also looked into clays in terms of drug delivery, and there's a good body of literature on what they can aggregate and pull into the inner layers for time release. And so that's another body of work that's looked
True.
at clays.
Yeah, we'll take a look at clays.
Well, I'll be happy to reach out. There's
please
much
do.
more to discuss.
Does anyone else have any other questions or comments or?
I see Ethan joined the stage. Ethan, did you want to add something?
Oh no, I already added to Serena's question. Yeah, it was just I guess I could add to a comment to Wise rather than go. I think the pyrimidines and the purines separate from the whole nucleotide would just also, it might, if you, seems like it would be plausible that you could find a shorter peptide sequence that might join up with those, in which case you would also have perhaps a more likely scenario to back up into creating nucleotides rather than hoping for the nucleotides to show up from somewhere else and then figure them out. So that was just kind of my.
Well, yeah, what's cool for just the purines and pyrimidines, and by the time they get the sugar and the phosphate together, they pretty much have to be at the edge of the clay.
Yeah, it's a good thing.
So and so that there's in terms of how they then condense, you're starting to build your RNAs or your so there's that. Go ahead.
No, I'm just curious. So you're proposing that there is within the clay essentially a gradient of chemical reactivity.
Yes, in the sense that, and that's been a property that these the pharmaceutical and the cosmetic industry have exploited. The natural clays there tend to be in the phyllosilicate layer. There's impurities, and so they're generally positive charges that accumulate, and so that will select for um, you know basically flat aromatic systems that interact well, or positively charged ions. And there's definitely a body on ion selective clays. And but then if you bring in the phosphates on the edges, so the geometrically will naturally segregate. But then in the swelling clays, they go through a transition where there's a monolayer of water in the dry state and a bilayer when it swells. And that's also has to do with the balance and charge imbalance in the clays themselves. So montmorillonite is like the canonical example of the swelling clays. And I was studying, those. But depending on what types of charges are in the phyllosilicate directs the the chemical potentials in the inner layers and what affinities different chemical species will have. And they don't covalently bond to the clays in the inner layers. There's hydration, so they're in essence in a two-dimensional sort of planar. Their mobility becomes two-dimensional until they get to the edges. But then you have all kinds of surface chemistry that could be happening there. Throw in catalysts on the edges, and it's a rich environment if it can be chemically reined in.
Yeah, that sounds. I mean, that's the kind of thing that I think will add a next dimension to what we're trying to do. I mean, we have to an extent, the particles that we showed that form do create essentially a new environment. You have basically the aqueous solution, and now you start to create these particles where different things happen. But actually, using more a template approach, and if clays inherently have some of these properties, I think very, very likely interesting things will happen if you just add them, especially if you have I think what you're saying is you have a confined area, perhaps between the clay layers, where covalent chemistry is not likely to happen, but then you diffuse towards the outside where you start to see new chemistries happening. And things like phosphorylation are super interesting. Also, phosphorylating amino acid side chains. I mean, it would be a very nice thing to show.
Yeah, it's a very very rich area.
And so, Serena, does this also have the effect, it sounds like, that it would concentrate the reagents
Yes,
in that area from,
yes, it
being
would,
much more, yeah,
and
yeah, because that would
that's
be really critical, at
well,
least
and that
from
was
like
the
the
interest in the
primordial spruce. Yeah, yeah.
Yeah, that was the
Because
interest from the pharmaceutical industry in um, uh, taking up, their drug molecules and concentrating it in the layers, because clays are also fairly non-toxic, and so you'd have a time release mechanism, perhaps and well, that was the interest, and there's a lot of work done on that.
I mean, to me, it's interesting in terms of the abiotic prebiotic situation, because I was actually surprised to have reread some of these primordial soups and realized that in addition to the amino acids, that there was some purines and pyrimidines being generated, but they were in tiny amounts. So I think a place that even if they built up over hundreds of millions of years during that perhaps analog actually happening of the
Well,
primordial soups, that they would actually have a way to make the reaction more likely if they were able to concentrate rather than being diffused
that is what
in the
attracted me
environment.
to
Yeah,
which is why I'm a clay river shore
yeah,
wet
yeah,
dry
yeah.
cycle enthusiast.
Yeah, I think you just give us some ideas for our. We have a whole bunch of summer students coming in who are looking for projects. So we it'd be pretty cool to see what happens in the presence of a clay. I think if you could, it's very likely that you'll find a complete redistribution of preferred sequences for sure. So yeah, we'll definitely follow up on that, and perhaps we
Oh,
can.
oh yeah, I'll reach out. Summer students, yay!
Yeah,
Yes,
we
I'll
have
definitely
no
reach out.
no shortage. After 2.5 years of pandemic, people are very happy to be doing things in person again after virtual summer student experiences. I think they're ready to go and do something in the lab. So yeah, um, any idea like that is that we can test over the summer is great. So yeah, love to follow up on that.
Yeah, one interesting thing to note at the start of the pandemic, some academics were clever, and they were they were clever enough to add the term coronavirus or pandemic into their research, and that allowed them to have lab access. I know a colleague of mine, a neuroscientist, he could not have access to his lab until very recently, and it was I think very infuriating because neuroscience, of course, always has all these participants and stuff. But I wish you all the best and all your students all the best. Hopefully, this was a fruitful session so that you feel perhaps compelled to recommend it to your students to
Yes.
come and visit us at Science Society, or perhaps you yourself would come by sometime and maybe even update us in the future of any discoveries or advances.
Yeah, I think we have to come back and talk about the clays after we've done the experiment. So
Wonderful.
no, thank you so
Wonderful.
much. The great opportunity for us to present our work and discuss it. So really appreciate it. Thank you so
Yeah,
much.
thank you so much, Ryan. And yeah, always come back. And yeah, this was such a wonderful discussion. I enjoyed a lot listening to it, mostly because Serena and knew would make this discussion also very interesting. And yeah, thank you so much for taking the time. And we really appreciate it.
Yeah, my pleasure. Thank you.
Thank you so much. It's been wonderful, and I look forward to further further clays.
Definitely, we'll follow up. Okay, thanks.
Okay. Yeah, thank you everyone also for coming and being here, asking questions, commenting in the chat. Thank you, Ankit, for sharing the papers and saying that this was fun. Thank you so much. Yeah, this was really wonderful. And yeah, if you like discussions like this, follow the club press on the little green house, and there you can check out what's happening next in our science society. Tomorrow we'll have Dr. Guo. He will talk about dogs and how they recognize dog and human emotions. Usually species only recognize like in a species their emotions, and dogs are quite special in being really good at recognizing human emotions too. And then we'll have Dr. Cheng. She will be talking about using molecular orbital based machine learning on Saturday evening. And yeah, and on Sunday we usually have our weekly recap rooms where we summarize what we learned throughout the week in an hour. So if you missed any of the guest speakers' rooms, we will share the links to their papers and like in around 10 minutes or so summarize what we talked about. So it's a nice way to like recapitulate what we did, and it's kind of rewarding for us because we realize how much we learned in this week. So yeah, thank you so much everyone.
And if
And
I may, just
yeah,
one comment before we quickly close. And if you are yourself a researcher, student, professor, industry leader of some sort, you're paving the way in some sort of new innovation or discovery, please reach out to us, let us know, and we'd be more than happy to feature your work, your research here on Science Society. So please reach out to Katerina, Serena, or myself, or any of the other green bean folks on stage.
yeah, that was great. Thank you, Eric, for reminding us. Yeah. And okay, we'll close the room in 3, 2, 1. Thank you, everyone.
Thanks,
Bye.
everyone.